Hi, my name is Aishi, and today I'll be talking about second messengers. Now, what are second messengers? They're small, water-soluble, non-protein molecules that are involved in cell signaling pathways. Now, be careful, they're not the initial ligand, the initial signaling molecule that binds to the receptor in the reception phase. They're the molecules that help pass on the message afterwards. So two of the most commonly used second messengers are cyclic AMP and calcium ions. Let's first talk about cyclic AMP. It all started with Earl Sutherland when he found that epinephrine was able to cause glycogen breakdown inside the cell while epinephrine itself remained outside the cell. So then he began a search for a second messenger that was able to transfer this signal along from the plasma membrane. What he found was that levels of cyclic AMP, or cyclic adenosine monophosphate, would actually increase when epinephrine binds to the plasma membrane. So how does this all work? Well, first, epinephrine binds a receptor, and this receptor actually activates adenyl cyclase, which itself is bound to the plasma membrane. And adenyl cyclase converts ATP into cyclic AMP. However, phosphodiesterase then quickly converts cyclic AMP to AMP. Let's go through the structures of ATP, cyclic AMP, and AMP. So here we have adenosine triphosphate with the 3 phosphate groups, and that's converted into cyclic adenosine monophosphate, which is shown here, by adenyl cyclase. And you can see how the phosphate group is linked this circular fashion. And cyclic AMP is then converted into AMP. So cyclic adenosine monophosphate is converted into just adenosine monophosphate by phosphodiesterase, as shown here. Now, what is the role of cyclic AMP in the cell? Well, it serves to activate protein kinase A, which is a serine-threonine kinase. And as you remember, kinases phosphorylate things. So protein kinase A would phosphorylate certain threonines and serines on proteins. And how are cyclic AMP levels regulated? Well, they're activating and inhibitory G proteins that are activated by different ligands and will cause the increase or decrease in cyclic AMP levels. Now let us talk about calcium ions. Calcium ions are actually even more widely used than cyclic AMP as a second messenger. Usually there will be an increase in calcium ion concentrations that will initiate some sort of pathway or cellular response. And calcium ions are used in both G protein and receptor tyrosine kinase pathways. So calcium levels in extracellular fluid are actually more than 10,000 times higher than calcium levels in the cytosol. So cells can actually adjust the calcium levels in the cytosol very easily by just allowing the calcium ions from the extracellular fluid to flow in, and that would increase the calcium concentration in the cytosol. So calcium is normally actively pumped out of the cell and out of the cytosol into the ER, and sometimes into the mitochondria and chloroplast, and is done actively. So when you pump calcium ions outside the cell and into the ER, ATP is used. However, when you're trying to do such a thing, when you're trying to pump calcium into the mitochondrion, then you can simply use chemiosmosis for your energy source. Now let's discuss the pathway that calcium and IP3 function in. So first we have a signaling molecule, and this will bind a receptor. This can be either a G protein coupled receptor or receptor tyrosine kinase, as you mentioned before. And this will activate phospholipase C. Phospholipase C will then cleave PIP2 or phosphatidylinositol 4,5-bisphosphate, which is a plasma membrane phospholipid. It will cleave PIP2 into diacylglycerol and inositol triphosphate. So the diacylglycerol will actually remain attached to the plasma membrane, while the inositol triphosphate is able to flow through the cytosol. And both of these act as secondary messengers. So the inositol triphosphate can then go and bind a inositol triphosphate gated calcium ion channel in the ER, and this opens the gate up to calcium ions that are inside the ER, and it allows the ions to flow out. So the calcium levels will increase inside the cytosol.